Hi, welcome to the Radiation Research Society vodcast. I'm Allyson Burrell, and here today we're interviewing Allyson Koyen, and she's from Emory University, a graduate student studying small cell lung cancer. Welcome.
Thank you.
We would all like to congratulate you for the Marie Curie Award.
Thank you so much.
And we'd love to hear about your research.
Yeah, first of all, I'm super excited to be here at this year's RRS meeting. And yeah, so I'm a graduate student at Emory University, and I'm working in the laboratory of Dr. David Yu. So our lab studies the DNA damage response and how its dysregulation can be utilized for targeted therapies for cancer patients. And so specifically, my project focuses on small cell lung cancer. And so small cell lung cancer, as you'll hear about later today, is a disease with very poor outcomes. So most patients that typically present with the disease, it normally has metastasized to a secondary site. So it's very difficult to treat. And so in the clinic, they really rely on not only radiation, but mainly chemotherapy in order to treat this cancer. And so initially, small cell responds very well to the first-line chemotherapeutic agents, but ultimately it becomes resistant to the chemotherapy, and patients experience tumor resistance and then recurrence, and then ultimately they will succumb to the disease. And so that kind of explains the poor outcomes associated with small cell lung cancer. The 5-year survival rate is only 7%, and the median survival rate is 24 months. So particularly poor outcomes. I think it's one of the cancers with some of the worst outcomes reported in the American Cancer Facts and Figures. And so it's really important to not only understand how we can better treat small cell lung cancer, but also develop and targeted therapies for this cancer. There's currently no targeted therapy for small cell, so there's an urgent need to develop these.
Yeah. Is this a familial type of cancer, or is it more spontaneous, or a combination of both?
I believe it's more spontaneous, but so I think small cell lung cancer has a couple of characteristic mutations, mainly through the loss of P53 and RB, but and I know some of those can be familial, but I believe it is a spontaneous cancer. Yeah.
So what types of therapies are you researching in your lab that you're testing with the small cell lung cancer?
Yeah, so we're focusing on the first line therapies for small cell lung cancer. So they, in the clinic, usually use a combination of cisplatin or cisplatin analog with atopicide. So my project stemmed from performing 2 screens using siRNA libraries where you knock down a gene of interest in the presence of cisplatin treatment or atopicide treatment, and then measure how the cell viability changes upon knockdown of certain genes, and potentially identify candidates that can sensitize resistant small cell lung cancer cells to cisplatin or atopicide. Okay. So we're mainly focusing on those agents. I know there are other second line therapies available, but because the first line therapies do initially work so well, and that's kind of the standard of care when the patients present with the disease, we're mainly focusing on those, but definitely we could broaden the scope to the other second line therapies as well.
Are the second line therapies more radiation therapies, or?
Well, I know radiation can be used particularly at the site of the primary tumor, but the problem again with small cells that it's usually has metastasized to a secondary site. So you really do need the chemotherapy, but it's not really radiation related. It's more of a DNA damage focused project.
Right. Does your lab ever use radiation to investigate the DNA damage? I mean, separate than?
Oh yeah, no, we have. We're very interested also in the double strand break response as well. We've published many papers on that pathway as well, and use radiation. And actually, atopicide is a type 2 topoisomerase inhibitor, can function similarly and cause single strand, but as well as double strand breaks. So pathways that are involved in mediating atopicide resistance may also have some relevance for the response to radiation treatment. So, I mean, there is some crosstalk there. With the cisplatin, though, it's a little bit of a different story, but
yeah. Oh, that's really interesting. So how far along in your project are you? When did you start in the lab?
So in our first year, we do laboratory rotations. So I did my rotation back in 2014, and then I entered the lab officially in 2015 and started my thesis project. So, usually with the rotations, you work on something unrelated just to get a feel for the environment of the lab and see if it's a good place. It's really nice actually, because it's kind of like you get to know what kind of environment you would be entering into as a graduate student. And yeah, so my specific project I really didn't start until 2015. So, I'm entering my 5th year in my program, which is kind of getting towards the end, and I hope to publish on my project related to cisplatin resistance and atopicide resistance moving forward. So I'm really looking forward to finishing up. But yeah, it's been a great experience. I've learned a lot, and I feel like, especially from a professional standpoint, it's been great to come to conferences like the RRS and present my work and get some feedback on what's going to strengthen publication when I ultimately submit to journals.
Yeah, speaking of publications, so your research focuses on these different types of agents that you're testing on cell lines, or is there any animal work, or what? I mean, you said you were using the siRNA library.
Yeah, so it's mainly focused right now on cisplatin and atopicide resistant small cell lung cancer cells. Okay. But we do have a collaborator with access to patient-derived xenografts for small cell lung cancer. And so we're sort of weighing the options. Our lab does not typically perform animal studies, but we're considering doing that in the future, especially to lend some more rationale for targeting some of these molecules in the clinic. If we can get some preclinical data in some animals in these patient-derived xenografts, it would be really, really interesting, and push towards a potential novel therapeutic target, or even just a biomarker for small cell lung cancer patients. So, I mean, of course, that's the goal. Yeah. So, I mean, just working with cell lines, you miss out on a lot. You miss out on the response from the immune system. You miss out on like the pharmacokinetics and pharmacodynamics of different agents. It's really hard to definitively conclude if it would be relevant in an in vivo system. So it's definitely like one of the weaknesses of my current work, and it's something that we hope to expand on.
So do you think in the future, looking for biomarkers that are specific to being treated well with a certain type of chemotherapy, would that be something that your lab might look into?
Yeah, absolutely. So from the biomarker standpoint, even if we can't see a conference of sensitization when we target a particular molecule, maybe that molecule is giving us a hint as to what pathway is being upregulated in response to the chemotherapy, and so we can measure like the levels of that molecule in a particular subset of patients and see how they might respond to certain therapeutics in the future. I guess that's kind of the whole idea behind the biomarker, but it's still kind of a long way off at this point.
So currently there are no biomarkers that you know of?
Yeah, for small cell, probably are. I know for a fact that the molecules I'm working on have not been characterized in that way, so that's always a good thing. But yeah, I'm not super knowledgeable about some of the specifics. I mean, again, there's some characteristics, like, the protein I'm talking on today, EZH2, like, for example, it's highly overexpressed in small cell lung cancer, like across the board. And, that's, of course, like, going to be very interesting if we can target it therapeutically, because compared to the normal lung tissue, maybe where it's not as essential, it's going to be a lot easier to target in the context of the cancer, where it's not going to affect the normal tissue.
For our audience that's not going to be here for your talk, do you want to give a little synopsis of what you're speaking on today?
Yeah, so today I am discussing the role of EZH2 in small cell lung cancer and how it mediates cisplatin resistance. And I found a novel role of how it's functioning through the nucleotide excision repair pathway. So if you're not familiar with cisplatin, which is a little bit different than radiation treatment, what it does is it forms crosslinks on the DNA, and normally this can be repaired by a number of pathways, but predominantly the nucleotide excision repair pathway. And normally what happens is the machinery will come in and scan the DNA for crosslinks, bind to that crosslink, and then unwind the DNA through helicase functions, and then exonucleases will come in and perform an incision of that crosslink, remove the area of DNA that has been crosslinked by cisplatin, and then polymerase will come in and fill in the gap and virtually repair the DNA. So that's the idea of the pathway. And so our lab found in a screen in small cell lung cancer that when you knock down EZH2, you get sensitization of cell lines that are normally resistant to cisplatin treatment, so they become resensitized to the cisplatin upon loss of EZH2. And so from there, we thought that EZH2 is likely mediating cisplatin resistance in some way. And because we know the NER pathway is important in terms of repairing cisplatin damage, we wanted to explore that role. But of course, EZH2 is a well-studied protein. It's been implicated in multiple DNA damage response pathways, cell cycle checkpoint dysregulation, proliferation dysregulation, as well as some roles in the double-strand break response. For example, the polycomb repressive complex number 2 does localize to sites of double-strand break. So we kind of wanted to see in an unbiased fashion how EZH2 might be functioning in the DNA damage response, possibly to explain how it's mediating resistance to cisplatin. And so we performed mass spec analysis and identified an interaction with an NER pathway protein DDB1, and we noticed that this interaction was occurring independent of DNA damage, and that we were also able to uncover an interaction with another protein in the DDB1 complex, DDB2. And so finally we looked at some readouts for the NER pathway, and that we found that silencing of EZH2 seemed to promote the direct target of the DDB1, DDB2 complex, which ironically is the ubiquitination of DDB2 itself, which promotes the completion of the NER pathway. So when you lose EZH2, it seems that it's destabilizing DDB2 in some way, but at the same time, it seems to be promoting the NER pathway, because when you lose DDB2 or DDB1, EZH2, or the two together, you also get an epistasis relationship where the knockdown of the two together seem to be sensitizing small cell lung cancer cells as well as other cell lines to cisplatin or EV at a similar level. So, from a therapeutic standpoint, EZH2 seems to be sensitizing cells to processes that are normally repaired by NER, but also it seems to have more of a direct role in this pathway through stabilizing DDB2 in some way. So we're still actively investigating what the specific role is there, but it's interesting that now EZH2 not only has links to double-strand break response and cell cycle dysregulation, but now also nucleotide excision repair. So that's essentially, yeah.
So therapeutically, EZH2, if you were to upregulate it, it would help sensitize the cells to cisplatin?
No, so
Sorry.
you want to down. So we're using
down.
siRNA to deplete the EZH2,
Oh, okay.
and then also you can use an EZH2 inhibitor. And actually, it's
All right,
really
that
exciting.
would be a lot easier.
Yeah, well, it's really exciting too because a lot of there are several EZH2 inhibitors that have been developed to inhibit its catalytic activity specifically. And some of them are undergoing studies in phase 1, phase 2 clinical trials. So, if these drugs are successful, they could potentially be repurposed for small cell lung cancer.
In conjunction with the cisplatin.
In conjunction, yeah, with the cisplatin, and yeah.
That's exciting.
That would be really great.
So you're working now on small cell lung cancer, but prior to graduate school, you were a postback at the NIH?
Yes.
And were you doing similar research, or what was your project?
Not at all. I was doing cancer research, but I was studying metastasis, actually, of osteosarcoma, and I was doing a lot of animal work, so totally different from what I'm doing now. But I was in the National Cancer Institute and basically working on a project that was more toward the side of clinical trials, where we were studying the role of translation initiation in highly metastatic osteosarcoma cells, and also studying how this could be targeted by a drug, rapamycin. So,
do you think for undergrads that are thinking about going to graduate school that doing something, an internship like a postback, is a good idea in preparation for graduate school? Do you think it prepared you better for starting a program or a project on your own?
Absolutely. So I guess I started as an undergrad on research, and I was working in a lab at Carnegie Mellon, but I was highly supervised by a postdoctoral fellow, and with only limited hours and not working full-time in the lab, it was really hard to get a sense of what effort is needed to drive and what creativity is needed to drive a project to completion. So when I entered the postback program, I had some experience in cancer research, but it was very tangential. I knew I was interested in it. I knew I wanted to pursue graduate work in that, but I had been working in Drosophila, and I had never really been exposed to cell culture, anything like that, which is a very important skill in the field of cancer research. And so, I learned a lot. I was working in the lab full time. I was driving the project on my own. I got a lot of independence very quickly, and was coming up with my own ideas and testing those out. And I think entering graduate school, it's still a training environment. People are still willing to take the time and teach you the assays and show you the ropes, which is really nice. But I felt very confident coming into graduate school that I knew what to expect out of working in the lab and driving the project to completion, and also just confidence in my own ideas, because towards the end of my postback, I helped prepare a manuscript for publication, and I kind of understood what that process was like, and I think it was just really excellent preparation. Now, being away from coursework for a couple of years was difficult, so coming back and immediately having to start taking exams and things like that, maybe I wasn't as fresh as if I had been coming directly off of my undergrad, because I had taken 2 years off just to work in the lab, not taking any classes. But once the coursework ended, I feel like I'm really in my element, and everyone has a different philosophy on how to train graduate students. You can find an advisor that's very hands-on or very hands-off, but I think in the end, it's your career that you're working for. You have to be proactive, you have to be independent, and I think working at the NIH just really taught me those skills, and just how to come up with my own ideas. And then, so really, when I talk to my PI, it's what I hope is that it's more on the level of colleagues, as opposed to me just asking, tell me what to do.
Right.
So that I feel like of course I'm not completely done. I'm not graduated. I'm not I'm not a faculty or anything like that, but I feel like, more confident, more prepared, and I had a really fun time doing my rotations too. Like, starting on new projects, I felt like I could adapt really well when I entered those labs, and could pick up on things faster because I was kind of familiar with juggling different kinds of science. Like I went from Drosophila work to metastasis work, and then I was put in all these different environments, and I was able to pick up on it really fast, as opposed to the very first time I was entered my Postback lab, and I was very unfamiliar with the research, and it took some time to acclimate to. But I think the more you switch fields and the more different fields you become exposed to, you start to see patterns and how to answer some of these important questions. And I really just I enjoy learning different ways to address questions, and I think my background has helped me a lot, because I never would have thought I would have ended up in a DNA damage response lab, but I enjoy it. I enjoy solving all kinds of puzzles, so it's been really fun.
I think that's really good advice for graduate students, is that you don't know exactly, you might have an idea of what kind of research that you want to do when you start out, but you never really know where you're going to end up. I also started in cancer genetics and ended up in radiation oncology and radiation biology, and it was going through rotations and experiencing different labs that made me make the choice to try something completely different than I had originally set out to. Yeah. Even cancer biology wasn't my original pathway, and but I find it, obviously very interesting now. And let's see. So moving forward, since you're coming towards the end of your graduate degree, so you would like to, it seems, planning on doing a postdoc.
Yeah, so
because you love research.
I really do enjoy the bench work. I know I can
tell by the way you talk about it that you're very passionate.
Well, it's exciting, especially when you make a big breakthrough. I feel like, just jumping up and down and celebrating. Of course, the work is not easy, and it's incredibly demanding. So, if I can keep it going, I would love, I love the flexibility of academia. I love the creativity that goes into it. I'm also considering, alternative career options. I think it's always smart to consider alternative careers, particularly because academia is so competitive and lifelong postdoc positions are not ideal. I think I'd also really enjoy industry, because it would combine the bench work aspect that I love, and then also the translational components of my research that I also enjoy and have a lot of background in. But academia has been great. It does involve a lot of sacrifice, long hours on weekends and things like that. But I also think it's a lot of fun. You're testing your ideas out, and it's just so creative and exciting. And when you find something, there's no cooler feeling.
Have you thought about what kind of postdoc you want to do? Do you think you're going to stay with a similar project to what you're working on, or are you going to take the same perspective that you took coming out of your postback and into graduate school and try something different and learn a new field?
I do enjoy learning about new fields, for sure. So I think I'm going to take that aspect with me at the next stage, but I think I do want to keep an aspect of my previous training with me, because I think it's important in a postdoc to come in with your own project so you can transition to independence. So I think bringing some of my own expertise to a new question would be my ideal scenario, I suppose. Now, how that's going to manifest, I'm not entirely sure. It's going to depend on where positions are available and so on. I'm really enjoying studying the interface of chromatin biology and the DNA damage response. That's an area I've considered, but I know also in today's day and age, like bioinformatics has become really important, and finding some way to pick up some training in that area would also be really interesting. But I do think I want to stick with the cancer research.
Thank you so much, Allyson, for coming and speaking with us. It was a pleasure to learn about your research and and hear your insights about graduate school and all advice to give to future graduate students as well.
Thanks.
Yeah, congratulations. Thank you.